So the last time we started talking about homeostasis and hormones. We talked about homeostasis in general, what homeostasis is and what it does and how it works. Some examples of where homeostasis is an action in the body to keep things stable, such as body temperature and blood glucose concentration, the water potential of the blood. Then we went on to talk about the systems that are involved in coordinating homeostatic responses, which are the endocrine system and the nervous system. So the endocrine system involves the release of hormones. The nervous system is about electrical signals or impulses. And as we will start off with today, right, the endocrine system is also for the most part under the control of the nervous system. Right? The endocrine system is contains glands. They're called ductless glands. They release hormones directly into the blood. They don't have any vessels or tubes through which they travel. It's just secreting it into the tissue fluid, and from there diffusion into the nearby capillaries very close by. They can be proteins, they can be steroids, depends. Protein hormones can be ADH, antidiuretic hormone, which we will study in detail. Steroid hormones are the sex hormones, testosterone and estrogen and progesterone. And they're called endocrine because the other glands that have either ducts, or if they don't, but their secretions don't go directly into the blood, so they go into a cavity, right? Called exocrine glands. An example are the salivary glands in the mouth. So, right, saliva is released into the mouth. It's obviously not going into the blood. And glands can be both endocrine and exocrine, such as the pancreas. So it secretes digestive enzymes, which is its exocrine function, and also secretes hormones, insulin and glucagon, depending on the blood glucose concentration, how it needs to be managed, and that is its endocrine function. So with that recap, today we will start off with talking about hormone release systems. So what causes a hormone to be secreted by the endocrine glands? And as I said, they are for the most part under the control of the nervous system. So if an endocrine gland receives a nerve impulse, it will secrete its hormone. An example is the adrenal medulla, of the adrenal glands, which secretes adrenaline. So adrenaline is a hormone. It is known as the fight or flight hormone. It is released in conditions of stress. By the adrenal glands. And so what happens is the adrenal glands are stimulated by the sympathetic nervous system. And the sympathetic nervous system stimulated by sympathetic nervous system, and the sympathetic nervous system is the one that controls our fight or flight responses. So if you are in a condition of stress, right, a signal goes out, an impulse goes out to the adrenal glands, and they secrete adrenaline, which go to their target organs, so the lungs, right? And so the adrenaline, right, target organs are lungs, muscles, heart, eyes, right, and it will cause an increase in blood glucose level, right, increased breathing, increased heart rate, increased muscle contraction, again fight or flight, whatever you choose to do, right, it will cause your pupils to dilate and all those responses. So there are many target organs for the hormone. Now there are cases where endocrine glands are stimulated directly. And directly, as in directly by what? So they can be, stimulated by other hormones or the chemical composition of the blood. Right, for example, the water potential of the blood could be a trigger, right, or the blood glucose concentration or anything else. Right, if there's too much carbon dioxide in the blood, that would, right, could be a trigger as well. So right, and so the relevant gland that detects that chemical secretes hormones to regulate the levels of that chemical, right? So relevant hormones secreted by glands, or I should say hormones secreted by relevant glands to regulate, right, the factor. So basically what's happening, if we there's a diagrammatic representation in the book, sort of a flow chart, and so what they've given is the endocrine gland in the center, if I draw it over here. So this is some endocrine gland, and what happens is, right, that the level of factor is too high. A factor or a chemical is high, and so what would happen is a hormone, right, the hormone that's supposed to control it, to bring it down, that will be secreted. So increase in hormone secretion, right, and that would decrease the level. And then the other way this goes is if the level of a chemical is low. Or a factor, let's call it. So it's supposed to be black. And so decreased hormone secretion and that would increase the level of the factor. Something like that. Now, the only problem with this is, this is a generalized flowchart. The only problem with this is, only takes into account, hormone secretion will be increased if the level of a factor is too high, and so the function in this case of the hormone would be to bring the level down, as it shows over here. But of course, there are hormones that will be increased, will have increased secretion if the level of a hormone is if the level of a chemical or a factor is low. So again, although it's not part of your syllabus, to explain this, if your blood glucose level is high, and so you will have increased secretion of insulin, that will lower the blood glucose level. And so that's this loop. But if your blood glucose level is low, now according to this, if you're talking about the same hormone, then yes, insulin secretion will be decreased, and that will, what insulin does is it converts glucose to glycogen, something we know. And so the level of insulin will go down, and so there will be less conversion of glucose to glycogen, and so that will help maintain, and the blood glucose level would go up again, because there's less conversion happening. But this also happens, when your blood glucose level is low. You have an increased secretion of glucagon, increased secretion of glucagon, which is, what this flow chart doesn't represent that, and that also helps raise the blood glucose level. Why? Because glucagon converts glycogen to glucose, converts glycogen glucose. So, in any case, as we talked about, there's a negative feedback system. Is that clear?
Yes, sir.
Any questions?
No.
Now, with that done, there is a mention of the pituitary gland, which it controls hormone release and has a pivotal role in homeostasis. So the pituitary gland is located in the brain, It has an anterior lobe and posterior lobe. So it has got two parts, and this guy is itself under the control of the hypothalamus. Which is obviously also in the brain. It's very close to the pituitary gland, it's just above it. And right, how it functions is that it monitors levels of chemicals or hormones and metabolites in the blood. Alright, so the hypothalamus monitors, because blood is obviously present throughout the body and very much so, right, in the brain, and so it monitors the levels of metabolites, hormones, and other chemicals in the blood. And if it detects something that is not in the set range, the average range that it should be, if it's too high or too low, whatever the case may be, and what it does is, right, it contains neurosecretory cells. And there are two of them, right? Neurosecretory cells 1 and neurosecretory cells 2. Right, and these guys, right, release, they secrete chemicals because from the ends of their axons. Right, which basically means they are neurons. Right, and if you're using the word axon, it's neurons, right? Nerve cells, brain cells. Okay. And so the neurosecretory cells 1 produce substances that either stimulate or inhibit the release of hormones, and it acts on the anterior pituitary gland. Anterior pituitary gland, right, and they can be either, they can promote the secretion of hormones, so those are called releasing factors. They promote hormone secretion and they're called releasing factors. Or they inhibit hormone secretion depending on what needs to be done, and those are called release inhibiting factors. Now the neurosecretory cells 2, right, they produce secretions that are stored by the posterior pituitary gland, and are released later as hormones. posterior pituitary gland and released later as hormones. Right. So, right, the anterior pituitary gland releases 6 hormones, the posterior pituitary gland releases 2. Right, what are the 6 hormones? They are the thyroid stimulating hormone, then you've got growth hormone, then there is the adrenocorticotropic hormone, then you've got the follicle stimulating hormone, and the luteinizing hormone. Now, all of these obviously have functions. Thyroid stimulating hormone controls secretion of thyroxine from the thyroid gland. The growth hormone, as the name suggests, stimulates growth of the body cells. The adrenocorticotropic hormone controls secretion of some hormones from the adrenal glands. The follicle stimulating hormone and the luteinizing hormones are both involved in the menstrual cycle and the implantation of a fertilized egg, right, in females. The posterior and I forgot one, prolactin, right? So prolactin, again a hormone present in women, and it stimulates and maintains the production of milk. Then you've got the posterior pituitary gland, and it releases only 2 hormones, so only comparatively, and those are oxytocin, again something that is involved in childbirth, so we find that in women, and the antidiuretic hormone, which is what we will be studying later on. Right, so the antidiuretic hormone is involved in regulating and maintaining the osmotic potential of blood. Now, next up, any questions?
No, sir.
No processes so far. I'll add just information. So we've got a process coming up now, and right, that is how hormones produce effects in their target cells. Now, in general, what hormones do is They activate secondary messengers through a signaling cascade. A signaling cascade basically meaning a series of chemical reactions activating different proteins in the cell. And what happens, right, is finally a protein that is a transcription factor right, and transcription factor is a protein that controls gene expression. So we know that the nucleus of the cell has DNA, and DNA is composed of nucleotide bases. It has sequences, and a certain stretch of nucleotide bases is called a gene because it codes for a certain protein. Controlling gene expression means controlling whether or not that gene will be activated and produce its protein through transcription and translation or not. So basically switching genes on and off. Right, so that is, and they activate a secondary messenger first by binding to receptors. So obviously that's the first step. So they bind to receptors, activate secondary messengers, and then finally, right, they either activate a transcription factor or however it works, and control gene expression. Now, there's slight differences depending on whether the hormone is a protein hormone or a lipid hormone, right, a steroid hormone. So if you have a protein hormone, right, it binds to receptors on the cell membrane. Why the cell membrane? Why doesn't it go inside the cell? Because, right, the cell wall is made up of lipids, and it doesn't allow proteins to pass through directly, right? And so you've got receptors on the cell membrane, and this begins a series of membrane-bound reactions, right? So the signaling cascade that I mentioned. And that ultimately leads to the formation of a secondary messenger, which in most cases is cyclic AMP, right? Cyclic AMP is made from ATP, right? So that's given in your book, made from ATP. Right, managed to squeeze that in. And then what that does is the secondary messenger, the cyclic AMP, right, also written as a lowercase c and then AMP, right? So that cyclic AMP, it activates various enzymes in the cell. Again, right, the continuation of the signaling cascade. And right, that affects the metabolism of the cell somehow. So how does it affect metabolism? Well, it activates, right? It binds to another chemical or another enzyme that goes into the nucleus. Enters the nucleus and acts as a transcription factor. Right, so the things it can affect cellular respiration, it can affect contraction of muscle cells or relaxation of smooth muscles and blood vessels, or the reabsorption of water, the permeability of cells to water. That is what ADH does, again something we'll talk about. So right, depending on the hormone and the target cell, it can have various effects. Any questions?
No.
Now, if you have a steroid hormone, right, it enters the cell directly because it's made up of lipid. The cell's membrane is lipid, and so, right, you're free to go through. So I haven't written that little bit over here, but I explained it, so do keep in mind that why the steroid hormone is able to enter the cell and the protein hormone is not. Right, so we know that we mentioned testosterone or estrogen or progesterone. These are the steroid hormones. They are made of lipids, right? They're steroids, and so they're lipid soluble. So they pass through the membrane, enter the cytoplasm, and act as the internal messenger stem cells. So instead of having secondary messengers, these guys are the messengers. And so what they do is that they bind to a receptor in the cell, in the cytoplasm. Right, form a hormone receptor complex. Right, and this hormone receptor complex goes into the nucleus and acts as a transcription factor. Right, and because it's, again, right, it's regulating gene expression, it's switching the gene on or off, and so RNA polymerase, transcription, mRNA, translation, protein, right, that whole thing. Is that clear?
Yeah.
Right, and that brings us to the end of this chapter, and then right to chapter 7C2. Now we do have time on the clock. I would like to continue. What do you say?
Sure, sir.
Okay. Alright, so 7C2, osmoregulation in mammals. Now, just like always, right, so we are dealing with science, we are familiar with scientific terminologies, and so if we come across something that looks new and looks difficult, and this is something the book suggests as well, this is something we have been doing in our classes. We did that photosynthesis and other terminologies, and so you break the terminology down. Right, so osmo over here comes from osmosis, right, which means something to do with water, right, the movement of water, the concentration of water. And regulation means, control. So control of water is somehow, right? And so that's what osmoregulation means. And so controlling the level of water in the blood or in the body of mammals. That is what we are going to study. Now, right, pretty simple. We understand that. Why do we need to control the level of water in the body? Because if there's too much water, if the water potential is high, in the blood, right, water will enter the cells, and the cells, because they are called mammalian cells, they don't have a cell wall, they will burst. That means they will die off, and that is detrimental to the body. If the water potential in the blood is too low, right, then water will exit the cells, they will shrink, and the contents will become concentrated. Right, and that obviously affects metabolic activities. So again, not good, right? Because they're not affecting metabolic activities in a good way, right? It's going to be detrimental effect. And so you want to keep the level of water constant, and so osmoregulation is obviously part of homeostatic control. Right, it involves the blood, right, and the kidneys primarily, right, and the hormone ADH. Right, saying the hormone ADH is kind of redundant, because ADH means antidiuretic hormone. So oh, by the way, even though we're not talking about ADH in detail, how it functions, right, that's the next chapter, so that's 7C3. But do you know what antidiuretic actually means?
No.
So, if I'm not mistaken, diuretic or diuresis means right, what's the word for it? Well, less concentrated, something along those lines. I'm forgetting the exact word. And so basically, when you are having a lot of water in the body, and you're frequent urination, and so right, the urine is less concentrated, there's a lot of water content, and so that is called diuresis, right? So antidiuretic hormone is basically, right, which basically means reducing concentrated urine. So that's basically what it means. So, right, pretty straightforward name. It sounds fancy because it's either Latin or Greek. So it's not English, and so it sounds like it's something like, no, you just translate it, and it's a straightforward translation. Right? Its name is its job. Okay. Now, Right, they go on to talk about how, right, mammals have adapted to control the amount of water in their body, how they retain water, and right, how much water is lost through urine, because they live on land and so the accessibility of water is limited, etc., etc. uh, right, and then they go on to talk about, right, how the kidney, they talk about how the kidney is involved in the homeostatic control of water, and one of the substances that is excreted, right, via the kidneys in urine. So it's not just water in the urine. So water is obviously the main thing, but there's something called urea, and there are also other salts, right? And so all of this goes through the kidneys. And so they talk about how urea is formed, right? And urea is formed in the liver. So what happens is excess amino acids, right? So when we consume proteins, right, they're broken down to amino acids. Only so much can be used. What happens with the excess, right? So glucose can be converted to glycogen, glucose can be converted to fat, etc., etc. Amino acids can't be stored directly, right? And so what happens to amino acids is that they go to the liver, where a process called deamination takes place. So I'm going to write by hepatic cells because, right, that's the term we use. And hepatic cells basically means liver cells. Right, so hepat, right, is the liver, and that's why if you heard of the disease hepatitis, right, so the hepat part is the liver, and the itis means inflammation. So the disease translates to inflammation of the liver, right? Or you heard of the hepatic portal vein or the hepatic artery and the hepatic vein, right? All also the hepatic means liver. And so they undergo a process of deamination. They are deaminated in the liver, obviously by the liver cells. And what that does is it takes oxygen, so oxygen is required. It takes the amino acids and it removes ammonia. Right? And so it takes 2 amino acids whatever amino acid it is, and you got oxygen, and what happens is you end up with a keto acid. So 2 of this guy plus 2 of NH3, right? So that's ammonia. And so how are we getting NH3 when we only have NH2? Well, if you just notice what's happening, oxygen's coming in and the position of the oxygen. So, right, the amine group along with the hydrogen at this position, those are removed, and so carbon, right, is 2 bonds short, and that's where the oxygen comes in, right? 1 oxygen atom from the oxygen molecule, it forms a double bond with the oxygen, and so now carbon has 4 bonds. It's happy. Ammonia is removed, and that is a problem, right? So we are still not happy. And the numbers are written in the brackets of the number of molecules involved, right? I've written them in brackets, not exactly how a chemical equation is done. Now the ammonia is a problem, right? So the ammonia is highly toxic and very soluble. right? And so it can easily dissolve in blood because blood is mostly water, and it is highly toxic, so it will cause damage to the body cells. And so what is done is, in the liver, the ammonia is fed into what is called the ornithine cycle. Now, the way they've given the ornithine cycle, they haven't given any intermediates or any chemical reactions or any steps of how this happens in the body. Right? The only thing they've given, right, is, right, you've got ammonia, which is coming from amino acids. So deamination of amino acids gives ammonia, and then it goes to a point where I'll just draw the circle over here going round. So it's a cycle. And so at some point, water is given out, it's lost, and then there's water coming back in, and then urea is formed, so that is removed. And then carbon dioxide is taken up, and water is removed, and then back to ammonia. And so as you can see, no chemical reactions, no the names of any intermediates or steps, right? Just they've basically just introduced something. Okay, this happens. Now, since they have given you the name, you obviously need to know that, right? But ammonia is put through that cycle, and it's converted to urea, and so that is of interest. Why is it of interest? Because urea is less toxic, not to say that it is something we want, comparative to ammonia. It is less toxic and less soluble, right? So it is less harmful for the body, and that is taken right in the blood. When it goes to the kidneys, it is filtered into the urine. So why did this come up? Well, because we're talking about the kidneys, so we talked about that as well, right? Obviously, not exactly osmoregulation, but yeah. So I guess I'll stop there, because the next topics are introducing the kidney, its structure, and then along with its structure, right, they keep describing the structure and the process of how water is initially filtered and then reabsorbed, and then more reabsorption, and so on, and finally the formation of urine. After that, the next chapter would be so we'll finally get to the point where we talk about how ADH affects reabsorption of water. That probably won't be this week, but right, I will obviously, right, so we have a class tomorrow, and so I will try and cover the functions of the kidney in formation of urine. Any questions?
No.
Okay, then right, do read up, stay up to date with the text,
Yes.
and I will see you tomorrow, inshallah.
Inshallah.